We have Aaron Goldman here with us, and I thought we'd get through some of the more specifics in general about DNA and DNA testing. Can you just give a quick overview about what is DNA and what are genetic variants or mutations?
Sure. Yeah, thanks for having me. So DNA is a long string-like molecule. When I say long, I mean very long. If you take all the DNA that's in one microscopic cell in your body, it'll actually stretch out to be about 2 meters long. And if you take all the DNA in your whole body, it'll actually stretch and wrap around the solar system twice. So it's actually one of the more amazing things that our cells do is we have machinery that package all that DNA, all that information into these microscopic cells. Every time a cell divides, it has to copy the whole thing. Every time you want to turn a gene on or off, it has to access the DNA. So it's a pretty amazing thing. So you might have seen in movies like Gattaca and in other media, the letters A's, T's, C's, and G's. So that's how DNA is usually represented with those letters. And those letters form a code. And that code is, it describes who we are and how we operate. Now, there are actually 6 billion letters of 6 billion A's, T's, C's, and G's within your DNA. And about 1% of that encodes genes. And the genes are basically the blueprints for proteins, and the proteins are what does things in our body. So the protein could be an enzyme that metabolizes a vitamin. It could be, a hormone receptor that guides development. It could be a neurotransmitter receptor that, modulates how we think and how we feel. So the DNA encodes the proteins go and do stuff in our body. And this is a quick overview, quick generalization here. Now, we're all human, but we all look different. So we're about 99.9% the same. So when you think of 0.1% of 6 billion, that's a big number. So we all have millions of differences in our DNA. And those differences, one of the most common type of difference is called a SNP, or a single nucleotide polymorphism, and that's basically a change of the letter, like an A normally, or in most people might have an A at a certain position. Some people might have a G at that position. So most of those changes actually don't have any clinical impact at all, or any outcome at all. But some of them can actually physically change the blueprint and then change the protein. And if it does change the protein, it could now be an enzyme, for example, an enzyme that works too slowly or too quickly, or it could not work at all. So that's what we're talking about when we're talking about genetic variants or mutations. We never like to use the word abnormal or mutant. It's really just a genetic variant.
Thank you for that. And can you elaborate a bit more about how do we go from understanding someone has like what you call the SNP to predicting clinical impact?
There's been a lot of study on DNA in the last 10 years or so, last 15 years. It's just been increasing dramatically because the cost of reading DNA has come down so much. So the field has sequenced like crazy, and we have a good understanding of how genetic variants relate to a certain outcome. And the way we know about this is from studies that are called genome-wide association studies. So the key word there is association studies. So for example, you could take 1,000 people that have a certain outcome. Let's say it's iron deficiency, and you take 1,000 people that don't have iron deficiency, and then you can compare what's similar amongst one group and different from the other group in terms of genetic variants. And that's typically how these genetic variants are discovered and reported. And those are the same genetic variants that we can look at in our genetic testing. So if the studies say that a certain genetic variant is statistically significantly are significantly statistically associated with a certain outcome, we can actually read the DNA now and provide that information in the form of a report to show you that, hey, you have a genetic variant that puts you at risk of iron deficiency. And then and then within the report, we can say here are symptoms to look out for, some guidance of how much iron you should get in your diet, and so on.
So how do you envision the genetic testing in the future based on what you've seen? And maybe add to, because some people are like, why all of a sudden, like, why am I seeing all these genetic companies all over the place? So it's a two-part question. Why are we seeing it now? Yeah. And what is the future?
Yeah. So the why now is pretty simple. As part of the Human Genome Project, which was a big project you might have heard about, it cost about $3 billion. So the first human genome to be sequenced cost about $3 billion. But part of that project was to develop new technologies, and then something called next generation sequencing came out in about 2007. And that's basically like going from a typewriter to a computer in DNA technology speak. So all of a sudden now we're able to sequence, the cost of sequencing has dropped down dramatically, and the whole field is able to learn more about how DNA works, but also we have the technology now to for companies to commercialize tests, to be able to take a cheek swab, take a blood test, take a spit tube, and read the DNA and report back on it. And what was the other question, sir?
The other question is the future of the genetic testing. And I'll share how easy this test is. So we'll have patients come in for their initial consult, and we talk about our health or our fertility audit, depending on what they're in for. And then they just do the swab right there, and it's sent off like it's seconds. It doesn't take very long. As long as they hadn't drank 30 to 60 minutes beforehand, they're good to go.
Right, exactly. Yeah, it's just a 30-second cheek swab, very simple. Yeah, the future, I think this type of testing is going to become pretty standard of care. I think we're going to look back 5 years from now, 10 years from now, and wonder how we practice medicine without this technology. I think one thing that I think is going to become very important is epigenetics. So looking at how DNA is methylated and how that changes how genes are being turned on and off. I think the microbiome is going to be key in the future. So the microbiome is the collection of bacteria that we have in our gut. Amazingly, humans are made up of more bacterial cells than human cells, and we're now starting to understand how different types of bacteria in the gut can predict certain things about health. And I think just looking more down the road, I think taking a whole omics approach. So not just genomics, which is DNA, but ribonomics, looking at the RNA, proteomics, looking at the proteins, and the microbiome as well. So I think it's going to be looking at all different, it's a big data issue, a big data problem, looking at all sorts of biomarkers. When people have their genetic information, they're more likely to stick to a program. So, for example, if you tell a patient, you should reduce saturated fat in your diet, they might do that for a month or two and then, slip back to old habits. Whereas if you have a genetic reason, if we say because of your DNA, you're at increased risk of metabolic disorder if you don't reduce your saturated fat, then people are much more likely to stick to that program. So 23andMe has done a really good job in terms of raising awareness of genetic testing. They spend a lot of money on advertising. So a lot of people, when you think about genetic testing, that's the first or the most well-known company that's out there. They do a great job with respect to ancestry testing. So they can tell you, maybe connect you to some long-lost relatives or tell you where you're from. What it's not, though, so while they do provide some health-related information, it wasn't really designed as from the ground up as a health-related test. Whereas Love My Health, our genetic test was designed as a health-related test. So, so I can give you a very specific example. The test for gluten sensitivity or celiac disease requires 6 genetic markers within the HLA gene. And of course, we test all 6 of those markers. Because 23andMe wasn't really designed to be a celiac test, they just so happen to hit 2 of those 6 markers, but they don't report on the other ones. So if you have, if you're negative, if you don't have markers within those 2, then 23andMe would report back that, you know, you're in the clear, with the fine print saying that we don't test all 6 markers. So there's going to be a lot of false negatives with a test like 23andMe when it comes to celiac, when it comes to things like they also do the BRCA1 and 2 test, but they're only looking at a handful of markers out of the thousands that are known to be clinically important. So you really have to, if you're going to be using the 23andMe test, you really have to understand what it is, what it isn't. It's really been designed for ancestry and data collection rather than, you know, as a lifestyle genetic test.